Today, our topic in bacteria is Archaebacteria, Cyanobacteria, Mycoplasma and Actinomycetes in Diversity of Living World, Unit 1, Chapter 1 in Botany. Archaebacteria, as I have talked about this Archaebacteria while taking the classification in three domains. The Archaea, it lives in extreme environmental condition. So they are the primitive prokaryotes. They are the first formed in the environment where it has extreme heat, extreme cold and extreme salt condition. Like that condition, this bacteria was there. So it is the primitive prokaryotes, and they are mostly chemoautotrophs. They, with the help of inorganic substance or organic substance other than the oxygen, they used to prepare their own food. That is chemical substance like organic acids or inorganic substances can be used by the chlorophyll to prepare their own food. That is called chemoautotrophs. This we have discussed in the nutrition topic. Unique chemical composition of the cell membrane. So the cell membrane has a unique chemical composition. Due to that chemical composition, it can able to resist the antibiotics and lytic agents. So it can able to resist. So some of the chemical antibiotics we are taking to kill the Archaebacteria, that bacteria fight with the antibiotics and it can able to resist and to protect it from the antibiotics. Examples: Methanobacterium, Halobacterium, Thermoplasma. Methanobacterium is a methanogen, so it needs methane to live the life. Halobacterium, salt condition it lives. And Thermoplasma, that is high, thermo means temperature, high temperature also it can live. And this is Archaebacteria structure. This was not in your book, so for simple to know you, this is the Archaebacteria. Okay. And next we move on to the Cyanobacteria, blue-green algae. So it was kept under algae, but nowadays the most of the characteristics are like bacteria, so they kept it in Cyanobacter or blue-green bacteria. So how old the Cyanobacter, that is stromatolites, reveals the truth. So they are the deposition form in colonies of Cyanobacter bind with the calcium carbonate. So they are covered with the calcium carbonate. The Cyanobacter is combined with, is joined or is mixed with the calcium carbonate. So that is called stromatolites. And you can find the geological age. So when it was originate in the world and what is the year, how many years it is old, you can find out. So 2.7 billion years old this stromatolites. So the fossil records that Cyanobacter helped in raising the level of free oxygen in the atmosphere. So the fossil records also giving that Cyanobacter can create more oxygen and releases to the atmosphere to increase the oxygen. So Cyanobacter is a blue-green algae. And how old the Cyanobacter? So that is stromatolitis, it reveals the truth. The deposition of calcium carbonate mixes with the Cyanobacter, and you can know the geological age, 2.7 billion years ago age. And the fossil record shows that this Cyanobacter that is help in rising the oxygen level in the atmosphere. And this is the stromatolitis, how the oxygen content is increasing and how they have done it. So this is just a picture of stromatolitis, Cyanobacter. And now Pseudomonas pudita is a superbug genetically engineered which break down the hydrocarbons. Okay, Pseudomonas pudita is a bacteria. It is a superbug. It was discovered by Anand Mohan Chakrabarty as an Indian scientist who lives in America. So he is an NRI. Okay, he created the what he created means he created the superbugs to digest the crude oil, camphor and octane to digest the crude oil. So that which is not degradable by any of the components. So but this bacteria can degrade this. So that was found out by Anand Mohan Chakravarti. And protein is a single cell protein derived from Methylophilus methylotrophus. Methylophilus methylotrophus, that is protein. It is a single cell protein which contains more number of proteins in a single cell. Okay, it is taken from Methylophilus bacteria, Methylophilus methylotrophus bacteria. Agrobacterium tumefaciens causes crown gall disease in plants, but its inherent tumor inducing principle helps to carry the desirable gene into the plant through the genetic engineering. So that is TI plasmid. So Agrobacterium tumefaciens, which causes crown gall disease in the that is in tomato, cotton, etc. Even brinjal, etc. It's like a cancer, how in human beings cancer, like that the cancer in the plants. But this bacteria, Agrobacterium, has a special plasmid known as TI plasmid. So that TI plasmid have means tumor inducing, but that plasmid was inserted into the plants like tomato and brinjal, and you can create an immunity to fight out with the Agrobacterium tumefaciens. So it plays a very important role in genetic engineering. And next, Dermis aquaticus. Dermis aquaticus is a thermophilic gram-negative bacteria which produces Taq polymerase, a key enzyme for polymerase chain reaction. So PCR. So Taq polymerase was taken from Dermis aquaticus. So polymerase, the PCR chain. Now we have to talk about this PCR because this is the one of the confirmatory tests to find out the corona test. So that for that the polymerase enzyme is taken from the bacteria Derma aquaticus. They may ask in 1 mark. In NEET also they will ask. Methanobacterium is employed in a biogas production. Okay, methanobacterium, the methane gas is the natural gas. For natural gas, instead of going for a natural gas, we can take the methanobacterium and we can get the biogas. Okay. And halobacterium, that is an extremophily, that is extreme condition it can live. So high salt condition it can live. Okay. And it exploits the production of beta carotene. So it can, due to the high condition of saltinity, this halobacterium can produce beta carotene. Cyanobacter or blue-green algae or Cyanophyceae. So the Cyanobacter is a prokaryotic organism, that is photosynthetic. So photosynthesis takes place. So because of that it is an algae. So and prokaryotic organism also. So the Cyanobacter is the primitive one for alga. And the different habitats, freshwater and few are marine. That is examples are Trichodesmium and Dermacarpa. So they freshwater and few are marine. That is the habitat. Habitat means where the plant can able to grow, where we are living in the environment, that is with a sophisticated house. So like that the plants where can able to live, that place is called habitat, that is freshwater and marine. And Trichodesmodium erythraeum, a cyanobacteria which creates red color. Due to this this bacteria only, Red Sea, you have studied the Red Sea and you have seen in the world map also the Red Sea, the color of the Red Sea, the color of the water is red in color due to the presence of this Trichodesmium erythraeum. Non-stalk Anabina, they are endophytic. They living an endophytic means they live inside another plant, like that is in cycas, the coralloids roots it is present. Okay. And it also, that is the Anabina is acting as a symbiotic association with Azolla plant. Okay. To fix the nitrogen. And the Glyocapsa, non-stalk, Syntonyma. So they are phycobionts, that is lichens. The phycobionts means they is along with the algae. So in a lichen thalli they are present. So okay, along with algae, and lichen is a combination of algae and fungi. So there they can be seen, this non-stack, Synthema and Glyocapsa. And you can see the chlorococcus. Coccus you have studied about it in the bacteria. Okay, chlorococcus, you have chlorophyll pigment here. And the next one is Glyocapsa. This is Glyocapsa, non-stalk, and spirulina. Next, Acinids. This is next one is fission, that is fission condition takes place in chlorococcus, and Acinids is seen in Anabina. This is Oscillatoria. So this and endospora is formed in Thermocarpus. So these are the forms comes under Cyanobacter, that is a blue-green algae or Cyanobacter. So salient features. So salient features, as we have talked about, this Cyanobacter is a prokaryotes, and it does not have any motility. It cannot able to move. Cannot able to move, and no reproductive, that is motile reproductive structures are absent. Motile means the reproductive spores cannot move. And the thallus is unicellular, that is single cell. You can see the single cell. During the fission it takes place into two cells. So that is unicellular in chlorococcus. That is colonial. Colonial means they live in the groups. So this one is Glyocapsa. You can see in the groups they are present. So that is in colonies. And filamentous, so like a trichome. So like a filamentous, a silk-like structure, a thread-like structure. So that is seen in non-stalk. And gliding movement in Oscillatoria. Gliding means it moves slowly, very smart. That is very softly it moves. That is called the gliding movement. So the protoplasm, here there is a central region called centroplasm. So here you can see the central, this portion is called the central part, that is called as centroplasm. And peripheral region bearing chromatophores is called chromoplasm. So at the sides you have the color. That color is due to the chromatophores, that is called chromoplasm. So center portion, the protoplasm is differentiated into two. The center it is called centroplasm, and the side and the margin place it is called as chromatophore, that is called chromoplasm. The photosynthetic pigments include Cyanophycean, phycoerythrin, and myxoxanthin and myxoxanthophil. So these are the pigments seen in this Cyanobacter. C, phycocyanin, C, phycoerythrin. So these are the photosynthetic pigments. Along with this, myxoxanthin and myxoxanthophil are also present. And the reserve food material is Cyanophycean starch. Okay, Cyanophycean starch. Next, that is, there may be some colorless cells in the terminal portion. They are called as heterocysts. This heterocyst help in nitrogen fixation. So the reproduction is only vegetative, and the vegetative cells produces acinids and homogonia, that is fission and endospores. You can see here the fission and the endospore formation. This is the endospore formation. So in this you can see the acinids and the endospore formation and that is fission. And the presence of mucilage, that is it was slippery. When you see the Cyanobacteria, that is on the rock side, the rocks are slippery, no? So when you keep your leg, it will be slippery. So you can able to fall off. So why? Due to the presence of mucilage around the algae or bacteria. So that mucilage makes the person to fall. That is due to the slippery one. So this is due to this. Therefore, this group is called as Mycoplasma group. So that is the thallus with mucilage. That group is called Mycophycean group. Next, the sexual reproduction is completely absent. Microcyst, that is Microcyst aeruginosa, Anabina flos aqua. So they causes algal blooms and toxins and releases. So they form in group form in more oxygen was there and it forms the algal blooms. Due to the over oxygen in the content, the organism living in the water will die, will affected. So the aquatic organism cannot able to take more oxygen. Due to that also the aquatic organism got affected and they got death due to the algal blooms. And most of them fixes atmospheric nitrogen that is for acting as an biofertilizer. Example, non-stalk Anabina, they are acting as an biofertilizer. And Spirulina is rich in proteins, that is it is a single cell protein. Even Spirulina tablets are used given for the skin diseases to enrich your skin also. So it is, these are the important points. And a prokaryote takes a joyride on polar bear. Echinocapsa monta, a cyanobacterium, grow on the fur of polar bear. So on the polar bear fur, the cyanobacterium called Echinocapsa montana. So it was on the fur, it grows on the fur. So that means where and all this prokaryote, that is with this polar bear move, the prokaryote take a ride. So they may ask in your NEET also. Echinocapsa montana, NEET, and even in 1 mark question also they can ask this. So Echinocapsa montana. Mycoplasma or Mollicutes. So Mycoplasma is very small, its size is 0.1 to 0.5 μm and pleomorphic, it changes its shapes, it won't have a definite shape, it changes its shape and it is a gram-negative microorganism. This was first isolated by Nocard and co-workers in 1898, where means in the pleural fluid of the cattle, that is in the lungs, that is a pleural fluid. There they had find out the this Mycoplasma, and it is affected with the bovine pleuropneumonia disease. So and this Mycoplasma doesn't have any cell wall. So it looks like a fried egg. So you can see this is a fried egg. It looks like a fried egg in culture. And it has DNA but low guanine and cytosine than the true bacteria. So it has guanine and cytosine are very less. Okay, the DNA contains very low guanine and cytosine than the true bacteria. So the next, it causes disease in animals and plants. Okay, what are the disease we will see? Little leaf of brinjal, witch's broom of legumes, phyllodes of cloves, sandal spike. These are the plant disease caused by Mycoplasma. Pleuropneumonia is caused by Mycoplasma mycode in the cattle. And this is the structure of Mycoplasma. It is surrounded by a cell membrane, and in the cell it has the ribosomes. The matrix is that you have seen as a pointed one. No, that is this one is the ribosome, and this structure is the DNA. So this structure is the DNA, and it looks like the fried egg. Okay, so this is the structure. They may ask you to draw the diagram of Mycoplasma. Actinomycetes or Actinobacteria. So it is also called as ray fungi. So why ray fungi means that is the mycelia grow like a rays. So the ray fungi, and it is an anaerobic or facultative anaerobic. Anaerobic means absence of oxygen, it will leave. And the facultative, presence or absence of oxygen doesn't matter. Okay, and it is a gram-positive bacteria. And the aerial mycelium, it won't be produced. The aerial only below the ground it will produce. Above the ground there is no mycelium. So think like that. Aerial means upper portion, that is towards the sunlight, aerial. And the DNA has guanine and cytosine in high. In Mycoplasma, they are very low, the guanine and cytosine. But in Actinomycetes or Actinobacter, high guanine and cytosine content is there. Example is Streptomyces. So Frankia is one of the symbiotic Actinobacterium. So it is a symbiotic, means it is seen in the root nodules, and it fixes nitrogen in non-leguminous plants. The leguminous plants means will produce, that is the proteins rich plants. For example, the pulses, red gram, black gram, green gram, groundnuts, in that they are called leguminous. Non-leguminous means they will not produce pulses. Okay, pulses. But also the plant need oxygen, that is nitrogen, not oxygen. The plant need nitrogen. That nitrogen can be fixed by this Frankia. Examples are ulnus and casuarina. Casuarina you will know. The casuarina tree. So in Tamil it is called Saukumaram. So they produces multicellular sporangium. And Actinomycetes bovis grows in the oral cavities and causes lumpy jaw. So the lumpy jaw, it can, in the oral cavities you can get some of the problems, no? A small infection will occur. So that is called lumpy jaw. And casuarina is also called as a false Christmas tree. Most of the Christmas time they will use this tree only for to put the, what you say, to put the Christmas, that is Jesus Christ has to born, no? So that type of a house can be created through the Christmas tree, that is casuarina. And this is the Frankia's structure seen in the root nodule. So this is a root, this is the root, and the root is surrounded by the Frankia. Streptomyces, it is also a Actinobacter, that is Actinomycetes, and it gives a good quality, okay, of medicine. Even when the rain comes, you will get a smell, no? That smell is caused due to the Streptomyces. So the earthy odor. So that is due to geosmin, an organic component present in the soil. Along with that is the Streptomyces was there. So it creates, after the rain, the soil smells. So the geomycin is seen in the Streptomyces bacteria. Due to that geomycin, the Streptomyces bacteria after the rain creates the earthy odor. Antibiotics, that is antibiotics like streptomycin, chloramphenicol and tetracycline are taken from some of the species of Streptomyces. And this is the Streptomyces bacteria. Thank you for watching the video. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.